Thanks for the great introduction. And Nikolai, thanks for doing all the heavy lifting on introducing my topic. I can breeze through the first third of this because I think you touched on just about every point I wanted to make. So we'll jump right into this. And here's just some disclosures on some of the work we've done. We have some funding from some industry partners to do some of this work. So our group is really interested in probably what most people are interested in, and that is to some degree the maintenance of homeostasis, and that's maintaining balance and when you're pushed around with a number of stressors. And one of the stressors that we tend to focus on is something that we can all relate to, and that's postprandial homeostasis, or how does your body deal with the mass influx of nutrients once you sit down and have a meal. And what always strikes me as interesting is the glucometers that we typically use in mice or even in people. They range from 0 to 600 if you're in the US, and for probably everybody else here in millimolars, from 0 to about 33 millimolar. Yet the glycemia in a random state is fairly tight, right around 5 millimolar or 90 mg per deciliter. And if you fast for a prolonged time, it only drops a little bit. And when you feed in a healthy state, it only goes up a little bit. And so this tight, narrow range, despite the potential for a dynamic range, means that there's pretty tight control over glucose homeostasis. And as many of you would predict, that's in large part due to the secretion of hormones from pancreatic islets. And so today we'll focus on beta cells and alpha cells. So to just set up the dogma or the canonical knowledge around these two hormones, this is really basic stuff. When glucose rises, there's a push on the beta cells to secrete more insulin, and insulin works on many tissues, including peripheral tissues such as muscle or fat or liver, to cause glucose uptake, and decreases hepatic glucose output, all in an effort to restore euglycemia. And the opposite is thought to be true in a fasted state. That stimulus drives glucagon secretion from the alpha cells, and glucagon liberates substrates from peripheral tissues like muscle in the form of amino acids or lipids from adipose tissue, and the liver will take these and use gluconeogenic processes to increase hepatic glucose production, or just liberate glycogen through glycogenolysis. And again, this is in an effort to restore euglycemia. So really, the way the field has looked at this since the discovery of insulin 100 years ago was that these two hormones act in opposition, almost in a push-pull, in order to maintain a very tight glycemic control. And so we come at this through the incretin axis, and just a brief introduction to the incretin axis. And by definition, really what this is the greater amount of insulin that's secreted when nutrients are given orally versus parenterally. And this was shown by Michael Nauk a number of years ago, about 25 years ago now, in a very elegant experiment. And what he did here was to take healthy subjects and give them an oral glucose tolerance test. And you can see the glucose rises up and comes down just as we would expect. And then he matched the glycemia in the circulation with intravenous, thereby skipping the gut. And you'll appreciate that when glucose is given orally, there's a substantially higher amount of insulin secreted, and that was attributed to incretin peptides that are secreted from the gut. So these are GIP and GLP-1. They probably need very little introduction. These are secreted from enteroendocrine cells found throughout the gut. And they have a plethora of actions throughout the body, but probably their most well-described action is the binding to their respective class B GPCRs located on beta cells, where they stimulate an increase in cyclic AMP and potentiate glucose-stimulated insulin secretion. And so part of the magic of incretins is they don't have very much effect on insulin secretion at low glycemia, which means that the risk of hypoglycemia is somewhat mitigated. They really require activation by a nutrient, in this case glucose, to stimulate the beta cell first, and then they can go on to potentiate that to a great degree. So why are we so interested in the incretin axis? Well, this is the same data that I showed you from Michael Nauk. This is actually taken out of the paper rather than shown in cartoon form. And so this was done in healthy subjects. And he then went on to do the same study, but this time comparing healthy subjects to those with type 2 diabetes. And you can see the fasting hyperglycemia, the greater excursion here, and the high levels of glucose at the end of this study. And when you look at the insulin in these two subject populations, you can see that there's, again, a 3 to 4-fold increase in insulin secretion when nutrients are given orally in healthy subjects. So this is the incretin effect here, but it's greatly diminished in people with type 2 diabetes. It's about half. So the conclusion from this is that the incretin effect in subjects with type 2 diabetes is substantially diminished, and that may contribute to the impaired insulin secretion in type 2 diabetes. So what's driving this? Well, there's been a number of hypotheses, but the literature basically centers around the idea that the incretin levels, what is being secreted from the gut, are normal or even potentially a little bit elevated, particularly with GIP. It seems to be elevated in the state of metabolic dysfunction. But it's the level of incretin receptors, the expression level, and potentially the activity on beta cell that is decreased. And this has been shown both in preclinical models and in islets from healthy versus type 2 donors. Now, the interesting thing is that the decreased expression of the GLP-1 receptor can be overcome by pharmacological levels of GLP-1. That is why we have drugs that target the GLP-1 receptor in clinic for treatment of type 2 diabetes. And this seems to be less true for the GIP receptor, that even very, very high levels of GIP fail to completely restore insulin secretion compared to normal people. And so that's sort of been one black mark against why GIP has not progressed as a therapeutic for type 2 diabetes to the same degree as GLP-1 receptor agonists. What about glucagon when we're looking through the context of both diabetes and then the incretin effect? Well, when you look at normal or non-diabetic versus people with type 2 diabetes, again, you can see the elevated glucose, the impaired insulin secretion, but they also uniformly show elevated glucagon secretion. This is what Nikolai was touching on the last talk. And you see higher levels, and in response to glucose, intravenous glucose here, where the levels drop in non-diabetics, they actually go up in healthy people. Now, the interesting thing is this is what you see in response to glucose alone as a stimulus. But here's some data from my colleague, Dave D'Alessio, and he did this, but he replaced it with a meal. So this is a mixed nutrient meal that resembles what we would typically sit down to eat at a lunch or a dinner, that has glucose, amino acids, and lipids in it. And you can see in healthy people, you even get a little bit of a rise here that is greatly magnified in people with type 2 diabetes. So this has driven the idea that type 2 diabetes is associated with hyperglucagonemia, and that potentially is driving the elevated glucose that we see here. So how do incretins play in here? Well, this is a recent review from my good friend Lori Baggio, who's back in Toronto, and she's using this figure to compare the effects of the two incretins that they have in common in the brain and on the beta cell and some of their unique effects. And one of the unique effects is in the alpha cell, where GLP-1 decreases glucagon secretion, and GIP is a little less clear. Some studies say that you might get a little bit of a decrease at hyperglycemia, but there are a number of studies that say that GIP actually stimulates alpha cell production of glucagon secretion. So in summary, one of the reasons we are interested in the incretin axis and why incretins have developed as therapies for type 2 diabetes is because they have this two-hit model when you look at the level of the islet. One is they robustly increase insulin secretion, and at least with respect to GLP-1, it decreases glucagon secretion. And using the canonical or dogmatic knowledge of what we think insulin and glucagon do, this would predict to have positive benefits for glucose homeostasis. So about 5 years ago, Megan Capozzi joined our group, and she was tasked with answering the question of what is the glucagon receptor doing in the beta cell? If these two cell types within the islet have a push-pull, and there is some evidence that products from the beta cell inhibit the alpha cell, well, what is the alpha cell doing when it's trying to talk to the beta cell? So she was tasked with trying to figure out what is its glucagon receptor doing in the beta cell. And one of the ways we get at this is with islet perifusion. And so I'll show a number of these experiments today, and I'll just walk through this first one. This is where we can take islets from mice or human donors and put them into chambers and dynamically change the environment within those chambers and measure the rate of insulin secretion coming out the other end. And so here's a very typical experiment where we go from low to high glucose. There's a first phase with an elevated second phase, and then give increasing concentrations of glucagon from low up to fairly high. And you'll see that glucagon, in line with the literature, is insulin-tropic. So it is glucose dependent, and at elevated glucose concentrations, glucagon will potentiate and stimulate insulin secretion. So Megan generated a mouse that's missing the glucagon receptor specifically in beta cells. And to our surprise, when we repeated this experiment in these knockout mice, it had no effect on glucagon-stimulated insulin secretion, which was somewhat strange if you take out the receptor for the agonist and it does nothing. And so when you get funny results, you dig through the literature. And many of you may be aware of this, because especially if you follow the development of multi-receptor agonists, is that the glucagon receptor, or the glucagon, is fairly promiscuous. And so this was best shown in some studies, some early studies by Franz Schut's group. And here we see that in hepatocytes, which only express a glucagon receptor, do not have a GLP-1 receptor, when he gives increasing concentrations of glucagon and uses cyclic AMP as his readout, the glucagon receptor antagonist nicely blocks this, and the GLP-1 receptor antagonist does very little. So everything is checking out. However, when the model is switched to primary islets, which express both receptors, now the glucagon receptor does very little, just like the data I showed you in the previous slide, but a GLP-1 receptor antagonist almost completely blocks the ability for glucagon to stimulate cyclic AMP in beta cells, raising the question of how important is glucagon signaling through the GLP-1 receptor in beta cells. Here is wild type islets with the GLP-1 receptor antagonist extended 9. And you can see here that we now block the majority of insulin secretion in response to glucagon, confirming the idea that glucagon signals primarily through the GLP-1 receptor. When we combine the two interventions, that is, we give the antagonist and beta cell GCGR knockouts, we completely wipe out glucagon signaling, suggesting that it does go through both receptors, but primarily through the GLP-1 receptor. And that's what this cartoon is showing you here. So now we have a term, alpha to beta cell communication, where glucagon, or probably more specifically pro-glucagon products derived from the alpha cell, utilize two receptors to stimulate insulin secretion in the beta cell. So here is a model to show the significance of this. Here we've now moved to amino acids, and as Nikolai nicely outlined in the previous talk, that amino acids are robust glucagon stimulants, and that's why we're using them here. Many years ago, we were interested in glutamine and arginine for other reasons, and have since moved to alanine. But now what we're looking at here is low to high glucose, and then stimulating insulin secretion with these two amino acids, and asking how much of this is driven by alpha to beta cell communication. To get at this question, we used mice that are missing glucagon in the alpha cell. So the beta cells are completely fine. We just knocked out the glucagon gene, and they don't make any pro-glucagon products. And you can see that the response to both amino acids is severely reduced, about 90% reduction. The interesting thing is the response to just glucose is reduced here. So even GSIS is reduced when we break alpha to beta cell communication by taking glucagon out of the alpha cell. And now you may say this is a funky mouse, you're dealing with genetically modified mice, but if we just give glucagon back, they respond fine. Or if we stimulate other GPCR ligands with GIP or GLP-1, they secrete normal insulin levels. And the advantage of this mouse is this is a stop flock. So we can take this out. We can take the stop flocks out in front of it and re-express glucagon in these cells. And when we do that with an adenovirus-free ex vivo 24 hours ahead of the experiment, we completely restore the insulin secretion profile to both glucose as well as the amino acids, and maintain the response to GPCR ligands, indicating that the beta cells are actually fine. They were just missing pro-glucagon products in the previous experiment. We can show this in human islets if we antagonize the receptors with a combination of a glucagon receptor antagonist as well as extendin 9. So here is just glucose alone in either the presence of the antagonist in blue, and we can reduce insulin secretion even at low glucose, and that is sustained through various levels of elevated glucose. We get impaired insulin secretion. And in response to amino acids, we can see that the response is also decreased when we don't have alpha to beta cell communication. So we published this data, so I'm going through it fairly quickly. And we came to the conclusion through a variety of mechanisms that alpha cells maintain cyclic AMP levels, and by doing so in beta cells, they actually maintain the tone of the beta cell and allow it to respond normally to other factors such as glucose, or even non-nutrient stimulants such as depolarizing factors like high potassium or KCl, or even sulfonylureas. But now we've got a situation where everything I've shown you has been in an isolated islet. We know that alpha to beta cell communication through these two receptors is quite important. But how do you compare that with some of the things that Nikolai was saying? Because we know that glucagon also has an important role on the liver and in hepatocytes to drive hepatic glucose production. So how do these two things balance against each other? So to do this, we took on a number of pharmacology studies where we were giving exogenous glucagon and basically asking how important is agonizing the receptors in the beta cell compared to agonizing the glucagon receptor in the liver. And so we did a study that many of you have seen. This is scattered throughout the literature, where we fasted mice and did a glucagon tolerance test. And here we gave glucagon and saw a nice rise up in glycemia relative to the control and stayed up with very little activity on insulin. This may just be because we've raised glucose a little bit. Now, remember that the activity of GPCRs and beta cells are glucose dependent. So the fact that we fasted these mice to bring them all the way down here to about 4 millimolar and then tried to stimulate them means that glucagon doesn't have a chance, because it's in a situation where it's unable to potentiate insulin secretion because we are at a low glucose. So we did a very simple experiment and repeated this, except we just didn't fast the mice. So now we have a situation where glucose is high, right? So now we're up here, elevated glucose. And when we do the exact same experiment, glucagon no longer raises it above controls, it actually decreases it relative to the controls. And that's because we're getting a robust insulin effect. So the hepatic actions of glucagon seem to be overcome when you're able to stimulate sufficient insulin secretion in the beta cells. And we have some tricks to show that this is not just glucose dependent. We can activate the beta cells with tolbutamide. So this mimics this will close the KTP channels and mimic the triggering effects of glucose, of high glucose. And when we do that, we get a low glycemia in the mice, but with an activated beta cell. And when we give glucagon, we get robust insulin secretion and a net decrease in glucose lowering. Again, because we have activated the beta cells. And finally, using genetically modified mice to just remove glucagon signaling in the beta cell, we knocked out both these receptors with a double knockout. We now, even at elevated glucose, only allow the hepatic actions to take place, and glucagon drives up glycemia rather than going down because it's unable to stimulate insulin secretion. Okay, so now we have the old version of the incretin effect, where nutrients through the gut stimulate incretins. Here I have GIP and GLP-1 could be considered here, and they potentiate glucose-stimulated insulin secretion. And what I've just described to you now is how the alpha cell is an important role in this context, where the alpha cell drives beta cell tone through pro-glucagon products on these two receptors. And in this last vignette of the talk, I want to talk about how does the gut talk to the alpha cell, and whether or not we can now tie in the incretin effect, which was previously from the gut to the beta cell, if we have another route where the gut can go through the alpha cell, stimulate alpha to beta cell communication, and contribute to postprandial insulin secretion. So this was Kim L's task, who joined the lab a couple of years ago. And she had the idea to focus on GIP, because we are an incretin lab and we look at GIP and other things. And we know that there's a GIP receptor robustly expressed on the alpha cell to about the same degree that it is on a beta cell. And she had this model where if GIP is needed to potentiate glucose-stimulated insulin secretion, maybe GIP potentiates nutrient-stimulated glucagon secretion in the alpha cell. And the nutrient she chose to focus on was amino acids because of their well-characterized ability to stimulate glucagon secretion. So here at this point, we had independently arrived at the same conclusion that Nikolai described, that we think alanine is the most important amino acid to talk to the alpha cell. And so now we've switched from other ones to almost exclusively using alanine. And here you can see we're measuring now glucagon in a perifusion system. This is at low glucose. And the ability for alanine to stimulate glucagon secretion is quite similar to what glucose does for insulin secretion. We have a first phase followed by a sustained second phase. When we give GIP alone at low glucose, we see a modest effect on glucagon secretion. And this is in line with what's reported in the literature. You get a little bit of an effect of GIP to stimulate glucagon secretion. But when we put them together, we get a robust synergistic effect where GIP greatly potentiates alanine-stimulated glucagon secretion. We have done this with other amino acids. So this is why I have amino acid-stimulated glucagon secretion. We just continue to use alanine because of its potency. In vivo, we can see the same thing. So these are wild type mice. Alanine and GIP alone give small effects on glucagon secretion in 10 minutes after we just dose them IP. But when we put them together in the same syringe, we get a synergistic effect on glucagon secretion, again, suggesting that GIP potentiates amino acid glucagon secretion. Kim built a mouse that missed the GIP receptor. We had knocked that GIP receptor out specifically in the alpha cells, and we can show that this synergistic effect, this threefold increase when we give GIP and alanine together, reverts back to what happens when you see alanine alone, suggesting that it's the GIP receptor specifically in the alpha cell that is needed for this effect. We can show the mechanisms to allow the synergy are because GIP and alanine evoke two different things. So here we're looking at cyclic AMP levels in alpha cells. This is work done with Matt Marins up in Wisconsin. And alanine itself does very little cyclic AMP, where GIP induces a large increase in cyclic AMP, kind of expected to be the case. And you can see that's true whether we give GIP before or after alanine. Where alanine is needed to stimulate calcium secretion, we see a nice robust effect of calcium here. GIP does very little to this, either given before or after alanine. So it really is looking very similar to what we see in a beta cell, where incretin stimulates cyclic AMP that can now work upon some of the pathways that are invoked by a nutrient. Okay, and then this is just showing that this translates from mouse to humans. These are human islets. We've now used a cocktail of amino acids here, which is just a little bit more potent in human islets. But GIP gives a nice potentiation of glucagon secretion when paired with amino acids, even in human islets. So the question is if we want to study this in the context of postprandial metabolism, we need to see whether this happens at high glucose. So does GIP in the context of a meal, so we did switch to a meal because we need amino acids in the system to unlock the alpha cell. At high glucose, which is probably inhibitory for the alpha cell to some degree, is GIP still able to potentiate amino acid stimulated glucagon secretion, initiate alpha to beta cell communication, and ultimately contribute to insulin secretion and the control of glucose homeostasis? So we'll start in islets. We have now moved to high glucose conditions, and I'll start with islets and then move to mice. So here we're testing this system, and our baseline is high glucose alone. You see first phase insulin secretion with the second phase. That's a little truncated because we have to stretch out the y-axis. If we give GIP alone in this context, you can see we get potentiation of insulin secretion marked here by the area under the curve, just in this area. If we give alanine alone, here's the effect of alanine to invoke alpha to beta cell communication. And if we look at the second phase, it's about the same what we give with GIP, maybe a little bit less depending on the experiment. And when we rope all the conditions together and we get all of this, we get a nice increase in insulin secretion relative to either of these interventions alone, suggesting that GIP can stimulate insulin secretion through direct effects on the beta cell, as well as indirectly through the alpha cell, as long as amino acids are present in order to trigger the alpha cell and initiate alpha to beta cell communication. Okay, so now I'm going to test this model by just showing you the area under the curve. So we're going to compare alanine and the ability for alanine to potentiate insulin secretion, all at high glucose in our various models. So here's wild type islets. Again, here's the effect of alanine above and beyond. If we do this in our alpha cell GIPR knockout mice, we completely lose this effect. If we do this by blocking out the beta cell communication pharmacologically, we lose this effect. If we genetically block alpha to beta cell communication, we lose this effect. And finally, if we take glucagon out of the equation completely, we lose this effect. So at least in primary islets, the model that GIP stimulates insulin secretion through both direct and indirect effects in alpha and beta cells is seeming to hold up. Okay, final data slide I'll show you here is whether this has any role in vivo when we start to look at the role of GIP in the alpha cell, given the multiple effects that both GIP and glucagon can have on glucose tolerance. So here's the experimental paradigm we have. We do an IPGTT followed by an OGTT, followed by a mixed meal tolerance test where the carbohydrate load is identical to oral glucose given. We just have the addition of amino acids in here. And the first thing we'll do in wild type mice is look at the area under the curve as we progress from left to right. Going from IPGTT to an oral glucose tolerance, we get more insulin secretion, not much of a change in glucagon. This is the incretin effect that lowers the overall AUC that we would expect. When we go one step further and include amino acids, even though we're putting more carbon into the system, we get a lower glucose area under the curve because we're getting more insulin secretion that we believe is due to a greater increase in glucagon and the inclusion of alpha to beta cell communication. To test this, we did the same test in alpha cell GIP receptor knockout mice. This has no impact on IP glucose tolerance because GIP is not involved in this system. In an oral glucose tolerance test, it also has no effect. Insulin goes up to the same degree, no change in glucagon because amino acids are not present to unlock the alpha cell and allow GIP to stimulate glucagon secretion. But in a mixed meal tolerance test, we have relative glucose intolerance because the mice are unable to mount the same insulin effect because they do not stimulate glucagon secretion. So this shows that at least in vivo, in response to a mixed nutrient meal that represents a normal feeding pattern or the normal components of a meal that we typically have, GIP requires activity in the alpha cell to stimulate glucagon secretion, mount the full insulin response, and provide glucose homeostasis. Everything I just said is now shown here in a really nice picture that Kim made, just showing that the effects of GIP occur in both beta cells and alpha cells. In a beta cell, we get direct effects that are glucose dependent. In an alpha cell, they're amino acid dependent. And when this whole system takes place, we have alpha to beta cell communication through two receptors that allow synergy with the GIP receptor and maximal insulin secretion and proper glucose tolerance in response to a mixed nutrient meal. Okay, I apologize again for the technical difficulties along here. And the last slide I just want to show is the people who did the work. This is our group now, and Megan and Kim were primarily responsible for collecting all of the data. We've had other lab members in the past that have been instrumental in this, our collaborators in funding, and I'm happy to take any questions.